Hello everyone, this is Mr. Tanner, and for this notes, we're going to be looking at how to use a Punnett square to predict the likelihood of a certain genotype in offspring based on the genotypes of the parents. And before we go into this, it is really important that you have your vocabulary down. This notes is going to be very vocabulary heavy, and if you don't know what dominant expression versus recessive expression or alleles or genotypes or phenotypes are, or homozygous or heterozygous, this set of notes is going to be especially confusing for you. If you don't feel like you have the vocab memorized yet, you have two options. You can either hold off on this until you've memorized it, or you can get your notes or your vocabulary set out while you're reviewing the notes so that you can reference any vocab words you're not familiar with as you're going through it. But again, the vocab is critical to your understanding of this material. So having said that, let's get started. So as I mentioned before, Punnett squares show the odds, that percentage chance, that a child or that offspring will have a specific trait based on the parent's genotype. And that means the mother's genotype and the father's genotype. And we're going to go through how we can uncover those odds for the next few slides. The first step here is to draw the Punnett square. So step one, draw the square. And that's what this is. So a Punnett square has 4 quadrants. So it's easiest to draw the rectangle first and then just do crossing lines through to get the 4 quadrants of that square. The next step is to know where the genotypes will be written with this square. So the father's genotype is written above the Punnett square, and the mother's genotype is written to the left-hand side. If you don't remember this, it's not important. As long as one of the genotypes is above the Punnett square and the others to the left, it's going to work out just fine. So the perfectly correct way is to put the father's genotype on top and the mother's to the left, but if this is switched, it will not change the results of the Punnett square. So a couple of conventions that you're going to need to know. I'm going to write these down here. Make sure you have these in your notes. So we have some conventions here. So one, the single letter code that we use for the alleles, so the 1 letter allele code, is the starting letter of the dominant trait. It's the starting letter of the dominant trait. So in this example, we're looking at fur color for rabbits. The dominant trait is black fur, so we're going to use the letter B. The recessive trait is white fur, but we don't use a W for that recessive trait because we use that same B since that's the dominant trait, since black fur is dominant. So that leads us to our second convention, which is the dominant trait is capitalized. So that's why we use the capital B here for the dominant trait. And the last one, the recessive trait, is lowercase. So even though the recessive trait is white fur, we represent that as a lowercase b. So this goes on, and what this Punnett square is showing is that the father's genotype is heterozygous. It has one allele for black fur here and one allele for white fur here, and so is the mother. The mother has one allele for black fur and one allele for white fur. So thinking about dominant expression versus recessive expression asks two questions here. The father is heterozygous, and so is the mother, so we can answer these both at the same time. But if the genotype is heterozygous, what is the phenotype? What color fur would the mother and father have? And the answer here is black fur. Black fur is dominant, and when you have one of each allele, the dominant trait is expressed. So the phenotype for these rabbits would be black fur. The next step is actually going about filling in the Punnett square once you have the genotypes placed above and to the left of that Punnett square. So one thing that's important to note is that capital letters, let's try that again. Capital letters are always written first. If inside of the square you have one capital letter and one lowercase letter, you always write the capital letter first. But you can change it around after if you lose your order. But capital letter always goes first. So to start, we just drop the letters above the columns down into each cell. So this B drops down here, and it would drop down right all the way into this bottom cell as well. So we have a B in each cell. Same over here. We have a this B is going to drop down here and below. So we have a lowercase b in each place. And then we fill the letters to the left in from the left. So this lowercase b goes here. This capital B moves over. The lowercase b also goes here. Capital B fills in. So when you're done, you end up with each cell having a full genotype in, where one of those alleles has come from the father, the red allele in this case, and the blue colored allele comes from the mother. So I'm going to clean this up a little bit so you can see what this would look like without the arrows filling it in. Let's look at this now without the arrows to muddy up the issue. In this top left column, we have big B, little b. In the top right column, we have little b, little b. In the bottom left column, we have big B, big B. And in the bottom right column, we have big B, little b. And you'll notice in that bottom right column that the capital letter went first, which is why the color order has been switched. So now we can look at the possible genotypes for any child. So this doesn't tell you all the child's will be, this is possibilities. And here's how we read those possibilities. Inside of this Punnett square, there's 4 genotypes total. So 4 total genotypes. And let's count the different ones that we see. So we have 1, 2 that are heterozygous. The order of the color doesn't matter. Those are both heterozygous traits where they have 1 dominant allele, 1 recessive allele. So 2 out of the 4 are heterozygous, big B, little b, which equals, as a percentage, 50%. 2 out of 4 is 50%. So we could say any child that these rabbits have has a 50% chance of being heterozygous, which would mean they would have black fur. We know that from the previous slide. The dominant trait is black fur, so these 50% chance the rabbits would be heterozygous for black fur. There is a 1 out of 4 chance, we'll do bullet points to keep track, that the rabbit would be homozygous dominant, that both alleles are for black fur. 1 out of 4 is 25%. And then there is a 1 out of 4 chance that the rabbit would be homozygous recessive, where both alleles are white fur alleles. And again, 1 out of 4 is 25%. And if you were to add the percentages up, they always have to add to 100% because there's, in the end, 4 out of the 4 full possibilities. So if I added 50 plus 25, that's 75, plus 25 again gives us our full 100%. So that you can just check your math at the end by making sure everything adds up to 100%. Now, this is the odds of the genotypes, but we could also look at odds of phenotypes. So genotypes are in black. Let's switch color here and let's look at phenotypes. There's only 2 total phenotypes. You can either have white fur or you can have black fur. Those are the 2 phenotypes. So we don't need to have all these different options below. It's simpler than that. There's still 4 options, but there's still 4 cells, I should say, but there's only 2 possible phenotypes. So if we look, this, and this all produce black fur. So 3 out of the 4, which equals black fur. 3 of the 4 will have black fur. And that percentage is 75%. So if you're just talking phenotypes, you could say these 2 rabbits have a 75% chance of having a baby with black fur. And the other one here is a 1 in 4 chance of having white fur, which is 25%. So the nice thing about Punnett squares is you can use them to track the odds of a genotype, which is just those 2 allele combinations, or phenotypes, which is the actual physical trait. We'll end our notes here with practicing filling out a couple of Punnett squares. I strongly encourage you to pause it before I start filling out and see if you can do the Punnett square on your own. It's a much better test of your understanding than waiting for me to fill it out. So pause the video, see if you can complete the Punnett square, resume the video, and then you can check your work from what I've done. So for this example here, we're saying what are the odds of a child being capital T, lowercase t, if the father has a genotype of capital T, lowercase t, and the mother has a genotype of lowercase t, lowercase t. Well, the father's genotype goes up top, so we do capital T, lowercase t. We'll pretend this trait, by the way, is tall, so this phenotype is tall. Capital T would be dominant for being tall. A lowercase t would be recessive for being short. Height is much more complicated than a single gene, but we'll just use it for now. So the father is heterozygous, capital T, lowercase t. The mother is homozygous recessive, 2 lowercase t's. So then we fill in the chart. Big T, little t, little t, little t, big T, little t, little t, little t. So the question is, what are the odds that the child has a heterozygous phenotype? Well, 2 out of the 4 possible genotypes are heterozygous, and we know that 2 out of 4 equals 50%. So the answer is that their child has a 50% chance of having the heterozygous genotype. One last problem here. This one looks at odds of both genotypes and phenotypes. Again, pause the video, try it for yourself, and then resume the video to check your work. So let's get started. It says brown eyes, capital B, are dominant. Green eyes, lowercase b, are recessive. That's our 2 alleles. It says the father is heterozygous. So the father must be, because we know our vocab, father must be capital B, lowercase b. And the mother is homozygous dominant. Both of her alleles are the same. She's homozygous, and both are capital B's. So once we have these laid out, we fill in the chart. Dominant trait, dominant trait. Oops. Try that again. Dominant trait recessive. dominant, 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 recessive. So there's our completed Punnett square. The first question is, what are the odds the child will be heterozygous? Well, there is 2 heterozygous traits. So 2 out of 4 equals 50%. 50% chance the child will be heterozygous. What are the odds the child will have brown eyes? Well, this gets into our phenotypes. So let's think about dominant traits. If you have a single dominant allele, you will have that dominant trait. So this cell is brown eyes. It has 2 dominant alleles. Same down here. These 2 cells on the right only have 1 dominant allele for brown eyes, but that's enough to express brown eyes. So in this case, all 4 possible outcomes for this child will be brown eyes. So the odds of having brown eyes is 4 out of 4, which equals 100%. If you need to go over this video multiple times, or if you need to reach out to me to get some help if you're not understanding the practice problems, please do so, because your next task is going to be to do 2 different assignments that are going to have you do questions just like this. It's important to be able to predict the possible outcomes based on the known genotypes. So this is using Punnett squares. Ask if you need help. Otherwise, good luck on your next assignments. Thanks for watching.